Might as well leave it blank. Might as well leave it blank. I know. Might as well leave it blank.
Oh, I didn't post the recording.
It's either in my dorm or what. Oh yeah. They found it. It's outside Wheeler. Take that shit down right now. Take this shit down.
That's my biggest fear. What store is on?
UMass 2028. No one would call that story. My U card is on there. Don't know what this happened.
Wait, I'm looking. Yes, bro.
Now everyone's gonna go look.
On him, is that his name?
How do I talk to this
I
guy?
believe we don't know that. No, not. What about his friend who clicked on the chat? The chat icon?
Yeah, I
Yeah,
tried.
that guy, and if you pull up the photo, I see everything like, I'm seeing this meme. Yeah, I know, I saw. Yeah, this kid goes like,
Hello?
you've never been with a Hispanic man before, have you? Like, you should like,
Just saw it.
There you go, okay.
Hello.
QRH. Yeah. Dang, less people showed up today.
Really?
Yeah, there was so many people yesterday.
I mean, I kind of assumed final exam review.
Yeah.
I spent like
Hold on, I'm gonna.
50.
Yeah, there was like 50 yesterday, I would say.
Really?
Okay, hold on, I'm gonna explain this question real quick and then
Go
we can
ahead.
start.
I might just move my seat for this. Wait, come with me over.
Okay, yeah, sure.
No, I can't work. Can you delete it?
Do you want to look at it?
Like, we tried to make a mega bed. Just
I just want you to like break in that and give them
I don't think I should have gotten all like just zero.
My brain is not.
Yeah, I don't think I would have given you all zeros here.
See, like, what the hell is who's like grading my shit? The same pen was the person who gave me zero on the question I showed you. So the guy who I brought whoever the fuck is still grading my shit for giving me consistent zeros on everything. They're like, why did you take it?
Okay, so it's weird because the way we grade, like, and like people take a chunk of exams and grade 1 videos.
Well, I also have.
Oh my god.
You guys know, I have a combination of like, you guys are like less strict on the way that I write things, like I have dyslexia, dysgraphia, and dyscalculia. Obviously, my writing is gonna be a little messed up, and I might not write things the way that I'm trying to word it. And I have that accommodation, I feel like no one's paying attention to that. How long is that?
You told Laura about that?
She knows that I have that accommodation. But I mean, you guys don't even grade it.
Laura did not. I did fine. Did you grade exam 1?
I grade exam 2.
Okay.
Like if I knew that, then
Like, I should have at least gotten something, because I feel like a zero and like a double, like, I feel like I had something correct. Because I know that you guys at least grade, but if I like get the wrong answer, but at least I have like the explanation that works with it. I should have gotten at least something.
I would like me. I would at least throw it as 0.5. I see like you're close in thinking, but you're still like wrong.
Because when I saw 65, I was like, where did I go?
Okay, can I go over this with you after, if you have time?
Yeah, so it can't be past 9:30. I'm trying to get the Japanese curry before I never do that shit.
No, I got you. This probably won't run the whole way anyway.
Okay,
Yeah.
perfect.
Sorry.
Japanese Worcester?
Yes, bro.
Well, walk me through the Worcester and do that.
It's like, you know that they close it at 10, so like,
I got stir fry that I'm trying to get. Don't worry.
it's bad, right? I'm gonna sit with you for 2 seconds.
Oh, I just saw your text.
Hmm?
I just saw your text
Oh,
about
was it?
candy. About candy. Yeah. I forgot to post a review session from yesterday, like the recording.
Oh.
So I gotta do that after this one too. I just started recording, so I remembered.
Oh, we're all good. Wait, did you record the last one?
I did.
Oh,
I just forgot to
fine.
post it. I just forgot to
Fine,
post it.
fine.
Yeah. Okay, whenever you're ready. We want to wait like a minute, maybe.
What time is it?
Sorry, I'd like to.
Oh, we could like start at 7:05.
Yeah, wait, I'm just gonna go through the tier just to make sure the questions came out good.
They had like the same.
Yeah, this is just a long question, so hopefully it's fine.
Oh, wait a
Because
minute.
it's the one you made, but like I made it two separate.
Okay.
Because I think on the doc it had two separate. It should be fine, because you can see big on here.
I'll like read it.
Yeah, we'll let him like digest it too.
Like, while they judge us, I'm gonna see if I like, even remember what I made.
Yeah, yeah. So that's good that you pulled this up. It's good.
No single top.
Yeah. Just like.
Oh, I didn't get to do the red circle thing.
That's fine. Don't worry about it. I just thought that it would be useful. I didn't even use it that much either. I did like maybe 2 or 3 slides last time, and 2 or 3 slides this time, so it'll be alright.
I felt like I was like very generic on like. There's something when it comes to what's it called. No. Where is it? Exokinetic,
Wait.
exergonic, and endergonic.
Are we doing that twice?
You had a slide on that. I noticed.
Oh wait, shit! Did I put in the wrong one? Well,
We can delete it, or like when it
can
comes
just
to it, I'll skip it for you.
no. Yeah, we can do that, or we can just talk about it twice because it's important. Yeah.
Yeah, no.
Or just like skim through, you know.
Yeah. Like we said earlier, exergonic.
Yeah.
And then just get to the next
Hey, it
one.
applies to both, right? Yeah. Okay. Which up?
I'm not sitting here.
Wait, what was topic 8 then? Oh, it's enzymes. So yeah, it actually does apply to both. So we're chilling.
Wait, what was yours? Isn't yours like cell respiration?
Yeah, cell respiration.
What is cell
So this is only I need it. Like, be honest.
There's something that ties into it. I forgot what it's called.
So this is all I had. So
Changing useful energy, spontaneous favorite, activation energy, so
yeah,
which one?
because like. Oh, discuss endergonic and exergonic, but we can do it twice. That's fine.
You want to read it or like? You just, like
Oh,
okay.
these? No. I said I'm putting them in the slides so they can look at
Oh,
it.
like when they okay.
Yeah, we don't have to read that shit.
Bro,
That'd be
bro.
crazy. That'd be crazy.
I said that I was like yeah, blah,
Just
blah,
like
blah.
yeah, it'll come up and be like, read this on your own time, but I included them. That's all. All right, we can start. Okay. All right, y'all, we're gonna get started. Settle in. Also, Niall, the mic's over here, so you're just gonna have to talk over here.
Wow.
And I'm just letting you know. So I'll move out your way because you're first. All right. Quick announcement. I didn't post the recording for yesterday's session, so if you missed it, it'll be up after this session. But it's coming. I didn't post the recording. Yeah.
Yeah.
All right.
We're all good.
Yeah, we're all good.
All right. I'm Niall. In case you guys don't know me,
I'm Sho.
You were here yesterday. You're fine.
Oh yeah.
All right, so basically what we'll be covering over today is topics 6 through 10. We're going to be alternating me and Sho through slides, and then please always stop us if we go too fast, or if we you just have any questions that you want us to go over. You can always pause and make time for that. Yep. So with topic 6, we're going to start off with cell signaling, signal transduction. Let's skip this.
Yeah.
So, to start off, the stages and outcomes of signal transduction. First things first, you always got to remember that you're going to start off with receptors, and that's like where reception happens. The cell needs to, the cell must be expressing the receptor for the incoming signal. So, if there's no receptor there available, the signal won't be able to get to it and go on into the cell. You also need to know that you need to have a receptor for your signal to go through because it just can't dissolve and pass through the membrane without one. It's kind of like a helpful buddy to get his message across, pun intended. Next is transduction. This is post-translational modification. You have covalent attachment and then phosphorylation. phosphorylation happens after that when a phosphate is added onto something. In this case, we're seeing kinases. Kinases are enzymes that are phosphorylated. It alters the protein shape, which I have a slide like after this that's going to help you guys remember. And it also alters the protein function. Also know that phosphates are an enzyme that dephosphorylates. Like, always, I like to keep this in mind that when it comes to phosphorylation, don't always think that something's going to be activated. Something could also be deactivated. And phosphorylation, as easily you can get a phosphate on, a phosphate could be taken off as well. Responses, changes in the existing protein function and changes in gene expression. Like I said, like when you turn something on or off, when something's phosphorylated, change of shape alters your change of function. I believe that's also on the next slide. Yeah. Like if you look at that first image, the unphosphorylated one, and then you look at the one to the right of that is phosphorylated. First it's kind of a little open, but when it gets that phosphate and becomes phosphorylated, it kind of clamps in together. When it clamps in together, what it originally could do has now been different. This could have been like, I don't know, like if you think of a light switch, when you flip the light switch on, the light comes on. That is one way, but if you add a phosphate onto the light switch, it's gonna flip the other way, and then the lights are gonna turn off.
Big thing here, I mean, I said it yesterday too, but change in structure is. There you go.
Mm-hmm. If you forget that, I like to think of Legos, just because I'm a kid in art. Every time you add a new block onto a Lego, it becomes something different. So that's just a fun way of thinking it. You could always like find your own way, because everyone thinks differently. So what works best for you is what's going to help you the most on your exam. Next is pathways. This image was on your exam 1 or 2. Do you remember, Sean?
2.
Exam 2? Okay. Understand how to read this image and how to do it, because if it pops up on your final exam and you look at it and you're like, oh, I'm fucked, like, you don't want that. You want to be able to understand everything from start to finish. So here you have on your left side is everything before the signal comes. You see the receptors aren't dimerized, but when you see the signal come on the right image, the receptors come together and they're dimerized. Once that happens, TK1 is activated. We don't know how it's activated. Just know that it becomes activated when the signal hits the receptor. After that, TK1 has the ability to phosphorylate TK2. And also something to remember is that phosphorylation from one kinase to another kinase isn't it passing on a phosphate like a baton. It gets the phosphate from like a floating ATP that's just inside. We don't know really where the ATP comes from. You just kind of know it's just floating around in there. You take that phosphate from the ATP and it puts it onto the next TK2. After TK2, TK2 has the ability to phosphorylate TFA. And then TFA, once that is phosphorylated, now has the ability to go inside of the nucleus and, activate all the stuff that goes on in there.
Yep.
So that's pretty much what you need to understand to get this diagram. Yeah. Let's see. Okay. Signal amplification and crosstalk. So signal amplification, I like to think of this as like, gossip. You have one receptor ligand that goes to a receptor and goes all the way into the membrane. And then once it gets in there, it has the ability to amplify into many different intracellular molecules. Think of it like I tell Sho a secret, then Sho goes and he's a snitch and just tells it to everybody else, and then everybody else tells it to somebody else. That's signal amplification. One person kind of spreads it out, and then it spreads it out even more. Another thing that you'd want to know is crosstalk. It's just good to be able to keep that in the back of your mind. That crosstalk is when in one pathway, if you kind of look. I don't know if you guys can see the mouse. I'm off my show. I'm exposing you.
Oh shit! Now you're chilling. That happened yesterday too.
Wait. Where's that? Okay. I'll do it slowly. You see over here in all this pink? That's one pathway. But if you look at these arrows, this pathway goes over to the green section. And crosstalk is talking between one pathway into another pathway. So if you're able to kind of draw a diagram kind of similar to this of you have one specific pathway and then like an arrow going to a different pathway, like all across the place. That's just a good way to kind of visualize it.
Yeah, I would just add like you have different pathways that do different things, but they reuse some of the same molecules, and because of that, those can go and interact with different pathways, which is just what crosstalk is.
Yep.
Yeah, let me see if I can get the pointer on. Might help you out. I actually don't know how to do it. Oh, okay, never mind.
Okay. So our first question is, the self-renewal signaling pathway in a particular stem cell type has an additional kinase, which is called TK3, and that targets TK1 and enhances TK1 activity. TK3 is produced from the expression of a gene activated by transcription factor A, also known as TFA. Will this be considered a positive or a negative feedback loop? Yeah.
It would be positive because it's enhancing the activity of TK1.
Yep. It's completely right. So I always note that when it comes to positive and negative feedback loops, it's something that's happening downstream that goes all the way back up the stream and enhances it in a positive way. And if it's a negative feedback loop, something that happens downstream that goes right back up, but it enhances it in like a negative way. All right, next question. Imagine that the example self-renewal signaling pathway is what signals for cell division in stem cells. You notice that these stem cells are no longer responding to growth factor signals and thus are not dividing. Which of the following situations could explain the behavior of the cell and explain the reasoning? Option 1 is there is a loss of function in the kinase domain of TK1. And option 2 is there is a mutation in transcription factor such that it cannot be phosphorylated by TK2.
Take a minute to think about it, and then talk with a friend, and then a group can answer, and we'll all give you candy.
A word?
Or we'll give you all candy, yeah.
If we get it wrong, do we still get candy?
No.
No.
You get candy. As long as you participate.
Alright, so where's that?
We'll give it like 30 more seconds.
Huh?
Could it be both?
I don't know, you tell me.
Unless it's which of the following, you don't say like pick as many. Is it an option?
You
think
have to
so.
explain for both. Or,
By
yeah.
myself. Like, what do you want me to do?
You want to borrow me?
Yeah, come
Yeah,
on.
you can answer the question for me.
Oh, no, I can't do that. Is this one?
So, yeah, it's this one, yeah. But
Wait.
the answer is both.
Oh, okay.
Yeah.
Hold up.
It was low-key a fire question you made. I won't lie. I don't remember making it. Or maybe you took it for something, but it's fire.
I think. Might have been a problem.
I think you made it yesterday, like the day we were working.
Yeah, I did.
Yeah.
But I blanked out for settings. Like, I
It's
forgot
true.
it.
It's good, though. It's good.
Thank you.
It's good, it's good. All right, 30 more seconds.
That's perfect.
All right,
Yep.
all right, everybody. So, can somebody or some group explain to me what they think about option 1? Could that explain the behavior? In the back. Yeah, could you explain?
It can't be phosphorylated by RCK, then you can't get the signal that would be
Yep, that's correct.
If you hit somebody.
I'm gonna come walk around. Don't worry.
Oh, okay.
Alright, you can press the first one.
Wait, hold up. What about the second option, though?
Yeah, you can answer the second one.
Yeah.
The second option would also cause the cell not to receive the cell signal because G1 phase.
Good.
That is correct.
Alright. Everybody good on topic 6? Any more questions? Yeah.
Let's say that the process or the signal transduction pathway was initially on and then the mutation occurred.
Okay.
Would we see a cellular response?
You'd see it initially, but if you're saying it starts, then the mutation happens. It'll go as much as it can, and then once the new proteins get made, it would shut off after that. Like there might be a little bit of a latent period. You know what I'm saying? Yeah. All right. Topic 7. Wait, let me just go. I forgot the name. Yeah. Again, these are your objectives for topic 7. Yeah, you guys can read them on your own. All right. You guys should be very familiar with cell cycle stuff. What phases there are, what happens in each phase. We'll go into the checkpoints as well. But to quickly recap, you have G0, G1, S, G2, and M. Can somebody tell me, aka read off the slide, what G0 does?
It's like a pause state where differentiated cells stay.
Yeah, that's good.
And it's like non-dividing.
Yeah.
Yep.
Can somebody tell me the big thing about G1? Or just shout it out. Growth. S. Well, okay, what does F do? There you go, replication. G2, growth phase 2. But also what happens in G2? Not quite. Mitosis is for M. Yeah, preparation. They do all the checkpoint stuff, make sure S phase went by smoothly. That's what G2 is. And then M, separate. Clear? Cool. All right, CDK is the biggest thing you need to know from this topic. If you don't understand this, you will not understand the rest of the topic. This is pretty much the biggest regulator of the cell cycle. So it allows for progression through each checkpoint. Its main activity happens in G1, but it's still needed in all the phases. And because it's such an important regulator of the cell cycle, it can be a big target for stopping the cell cycle when necessary. So the three regulators of CDK are cyclin, different kinases, and other protein inhibitors. So the different kinases are CAK, and then WEE1. Can somebody tell me how CAK and WEE1 interact? Take a guess. You'll get candy. Yeah.
Cyclin activates CDK so it can move the cell cycle forward.
Mm-hmm. And then how does WEE1 interact with it?
It is an inhibitory factor that stops CDK from going forward with the cell cycle.
Yep, that's correct. So basically they're both kinases, so they both phosphorylate. But WEE1's a good example of how phosphorylation can inactivate or inhibit something. So make sure you remember that. CDK it plays a big role in the G1 checkpoint. A lot of you could remember the G1 checkpoint as the point of no return. If they commit past this point, they will be committed to the cell cycle, and then obviously can't go out of it. If they're before the G1 checkpoint, they can still just not be in the cell cycle technically, and then go back to G0. But once a cell is ready to move on, CDK is dephosphorylated. The phosphate put on to CDK by WEE1 is then taken off. That's how CDK is activated, basically. CDK will then go on to phosphorylate RB, which is a different protein involved. The phosphorylation of RB will then cause it to release from E2F. E2F is a transcription factor. Please remember that. A lot of people say it's different things, it's DNA, it's whatever. It's a transcription factor. So make sure you say that on the exam. There will most likely be at least like a lot of questions on this. And the E2F's job is to initiate S phase by initiating transcription for a lot of proteins needed for S phase. That's kind of how it works. Anybody have any questions about G1 checkpoint? Cool. So S phase DNA replication occurs. One thing I'll mention really quickly, the word homologous chromosomes is a little confusing for a lot of people. This diagram may help, but I'll get a lot more into it in topic 10, which is later on today. But yeah, DNA replication occurs. You don't need to know how exactly it happens. You just need to know that this is what happens in S phase. The chromosomes and centromeres are duplicated, and the resultant product is homologous chromosomes, which is the two here on the right. Then after S phase, you have the G2 phase and G2 checkpoint. So again, G2 means growth, but the second time, it creates all the proteins you need for M phase, and the big thing is it checks for DNA damage. Any mutations, breaks, other kinds of damage, whatever, it just checks for that. And if there is DNA damage, it'll go in and repair it. Otherwise, it can't progress to M phase. Once everything's all set in order, it can pass the G2 checkpoint and go into M. But before M, or I guess this is during M technically, but yeah, spindle assembly checkpoint. Another big topic, you should understand how this flowchart sort of works. Cells need to have a way to sense that microtubules from opposing spindle pores have attached appropriately to the opposing sister chromatids, kinetochores. Does that sort of make sense for people? Like how it's important they attach properly? Cool. What happens if they don't attach properly and separate? What's the big word? Yeah, non-disjunction. So the cell will inhibit that segregation until the proper alignment happens, until the proper attachment. Before then, it can't separate. So spindles do arguably the most important thing during M phase, which is the actual separation. So it's important that that doesn't happen too soon. All of it needs to be aligned properly. Cool. Any questions?
And the spindles connect at the centromere?
Yep.
It's kinetochore?
Yep.
And the centrosomes are like the complex where the spindles come out of, right?
Yes.
Or is that centrioles?
Wait, repeat that one more time?
Yeah, one more time.
So the complex where spindle fibers come out of,
Mhm.
the little like L shape, that's centrioles, right?
What do you mean by L shape? Sorry.
You're talking about like the blocks
Yeah,
on like
yeah.
the either side, and then like the little, I call it like spider webs, like the
Mhm.
spindles come out. I don't know it off the top of my head, but I can look it up for you. One do P53.
Oh yeah. P53 is a transcription factor, as you guys know. It regulates cell response. Can somebody tell me if... Actually, wait, this is a question later on. I'll ask it in a sec. When damaged DNA is sensed, the ATM kinase is activated, which then phosphorylates P53, making it active. Once it's active, it can do two main things. It can either pause the cell cycle to fix any damage that occurs. So it upregulates the transcription of P21, which is a CDK inhibitor. CDK is, again, the thing that causes the cell cycle to go forward. If it's inhibited, it's paused. Or it can signal cell death. If it realizes that the damage is irreparable, whatever happens, it can just cause apoptosis. Yeah.
So P53 is a tumor suppressor,
Yeah. Well, yeah, that was. I have a question there, but you answered it, so good job. There it is. Yeah, tumor suppressor. So can somebody tell me what happens if there's too much cyclin or CDK?
Do you want?
Would the pathway happen too fast?
Yeah, correct. So if the pathway happens too fast, what could you tell me about the size of the cell?
There would probably be some problems with like taking growing too fast or not growing enough.
Yep, that's perfect.
Could it also like get too small instead of is it just overgrowth?
So yeah, it gets. I just didn't write it. Yeah, you're right. The cells themselves could be smaller, but generally there'd be overgrowth because there's be a lot of cells just there. Yeah.
If there's like an overproduction of CDK so that the cells are smaller than normal, because there's not enough resources, that would be stopped by G1 checkpoint, right?
Yeah, G1 or G2. Yeah.
Oh yeah, the answer to your question earlier, those like things on the end where the what's it called centrioles come out of, they're called centriole.
Centriole. Yeah.
Yeah, it's just because like there's 3 or 4 words that
Sound the
exactly
same,
the
yeah.
same.
Yeah.
It's like centromere, centrosome.
Mm-hmm. Nah, I got you.
Cool. Any other questions? All set. Cool.
You said P53 is a transcription factor?
Yeah.
Because
Yeah.
if it's
Yeah.
phosphorylated, it promotes
Cool. Back one.
P21.
Yeah.
Mm-hmm.
Yeah.
All right. Now for topic 8, enzyme function kinetics. You guys can look at this later. Quick basic stuff. An enzyme. Enzymes are proteins, and enzyme also catalyze reactions. Another term for this is saying that enzymes are catalysts. The enzyme on it has an active site, and substrate will bind to the active site. And then after that, there will be like a load of reaction that occurs, and then what will come out of that reaction is going to become products. I always like to remember that, like, in a business, the thing that you make, I pretend like that's my reactions, and the thing that I'm going to sell at the end of the day is going to be my products. So that's kind of my like mentality of how to think about it. And it also helps really well reading an energy diagram like this. Pretty sure this is the exact same one. Might have been on your problem set. I don't remember too well. But think about your activation energy. And always remember reaction goes to products. This little like hill is your activation energy. It's also made out of Gibbs free.
Yeah. It's
Is
called
that
Gibbs
good?
free
Okay.
energy. Yeah.
Okay.
Yeah.
I don't want to mess myself up. It's made out of Gibbs free energy. Let's
That's
see.
the delta G. If anybody's known. That's
Yes,
Gibbs free energy.
When it comes to activation energy and enzymes reduce the activation energy of a reaction. So between like the little, like this little hump and this big hump, which one do you think would have more enzymes in it being pumped into the reaction? Yep.
The top one?
You think more enzymes would be into the top one? So if I told you that enzymes make a reaction go by a lot faster, do you think to get to the end over here, do you think it's easier to like climb up this really big steep hill?
Oh, no.
Or you think the bottom one? Okay. So that's pretty much how you think of enzymes. Enzymes make like your life just a lot easier. You know, I'd rather go through this little hill than go all the way up this big hill. So the one that would have the most enzymes going into it would be the small one. I fucked up.
You're chilling.
Alright. Oh yeah, also always note that enzymes are catalysts. So if you get a question like this and Laura specifically doesn't say enzymes, but she says catalysts instead, just think of it the exact same way because enzymes are catalysts. Another thing you like to know is exergonic and endergonic reactions. I like to think of it as exergonic as like exer, so exit, energy is going out of me, and ender, energy is coming inside of me. I also like to think, because like me, you know, like I like money. So say like I'm giving like money away, right? I start off with a lot of money, so I start off with a lot of products. If I'm going to be giving that money away, I'm going to be broke at the end of the day. So I'm going to be really low when it comes to my products. Versus something that's like endergonic, where I need to take energy in. I'm really poor and I want money. So as much money as I can get, at the end of the day, I'm going to be richer and I'm going to have more products, more energy, more products, more money, me happy, you know? So that's one way you can think about it. And
Yeah.
like, oh, my phone.
What is like the Gibbs free? Like, why do we have to still worry about the delta G now?
Yes. So from the delta G, what you should know is exergonic. What Niall just said, energy exits the system. So when energy is exiting, the delta G of that system will be negative because you lose energy. You can think about it. The delta G is negative. Versus when energy is put into the system or added, delta G will be positive.
So is delta G just the change in the reactants, products over reactants? I thought delta G just doesn't change.
Yeah, so delta G of a reaction will never change. It's always going to be products minus reactants. But if you go back to the other slide, the enzymes is what changes the activation energy. Basically, how much energy you need to start the reaction.
Mhm.
Yeah.
Also, free energy, just think like something that's free just isn't going to change.
Yeah.
So that's like, just keep that in mind. Okay, so our question is, should we expect enzymes to have a specific affinity for their substrate and why? And then you guys can like take your time, try to answer that, and I'll ask the second one after.
Yeah.
Yeah, we should.
Yeah. Talk to your neighbors.
Oh, my fault.
Discuss. Yeah.
Wait,
I want that candy.
I got
I'll get
you.
You already participated.
I don't want busted ears.
What do you want? What do you want?
What do you guys have?
Twix, Milky Way. Milky Way.
All of them suck.
Or Snickers. Yeah. So if let's just say to quantify it, the reaction has a level of 10. And if it releases energy, it'll go down to like 5. So 5 is it's technically negative 5, right? So yeah, negative 5
Oh, so got all of it. Sorry, I was in
Smart.
We're on 8.
Yeah.
Right, yeah.
All right. Who's got an answer for one?
Yes, enzymes should have specific affinity for the substrate because each type of enzyme only catalyzes the specific reaction, so they need the correct substrate.
Yeah, correct. That's why they call it a lock and key mechanism, if you've heard that before.
Okay. Next question is, do you think that enzymes need to have affinity for a product of the reaction? Yes or no, and why or why not? Oh, wait, hold up.
Oh, sorry.
Did you say yes about that?
Yeah, you got it. You both can say it if you want. one at a time.
You both get candy.
Yeah.
You
We'll give you
don't
that.
wanna have affinity for the product, cause it's occupying a space. You wanna get rid of it as quickly as possible, so you can keep making more product. You do not wanna have a high affinity for the product.
I mean, they need to have some level of affinity because they can't release the product right away. They need to be able to keep the product so that the reaction is fully occurred and it can be released in a specific time when everything, like when the cell is ready.
I would say, yeah. I would say the most accurate answer is it needs some affinity. It definitely needs affinity for the substrate. The transition state will also have affinity because it's not completely made yet. But as soon as the final form of the product's made, it shouldn't have affinity so that it can be released and then goes to the cell and does whatever it needs to be doing. I think that's the best possible answer.
1, 2, all the way back.
Yeah.
Oh, my fault.
You want Milky Way?
Dang, that's all we have left?
Yeah, that's plenty. We gotta give him out like carefully now.
I don't know.
Oh, another one?
Back right.
Oh, my fault.
Nah, you're good, you're good. That's you though.
Alright. Alright, everybody good on topic 8?
We didn't talk about the graph.
Which one?
Michaelis-Menten graphs.
We're gonna have, those are not.
Well, yeah, okay.
For sure on the exam.
I know.
That's an open response. I
The
can
thing is, you
sense.
can remember, like, ice absorbs, like, heat, that's why it melts, so that's endergonic. Fire releases heat, that's why it.
Yeah, but what about like competitive inhibitors, non-competitive?
That one's fine, though, go and explain that.
We can get to Michaelis-Menten, like the enzyme stuff. What is it? The K50, KM? No, it's not. This is not Michaelis-Menten.
Wait, it's not?
It's this one.
Hold up. Oh, my fault.
Yeah. We'll get to Michaelis-Menten. We can do it at the end if that's cool.
I got a picture of that somewhere.
Yeah, I think there's a slide in there somewhere. All right. For now, cool on topic 8. For now, yeah, we'll get to Michaelis-Menten. Don't worry. All right. Topic 9 is cellular respiration. Again, this is like a pretty dense topic, so just remember all the steps. You're going to need to know the products of each of the parts of cellular respiration, so make sure you have those down before the exam tomorrow. Okay. You guys can look at that. Metabolism, generally, I'll go through it. More stable molecules have lower potential energies. Do you guys get that statement? Does that make sense? Why? Can somebody tell me why? You've been answering a lot of questions. Can somebody else tell me?
You find like a synonym maybe for lower potential energies? What do you mean by that?
What's that?
Can you repeat the statement?
Yeah, more stable molecules have lower potential energies. What does that sentence mean? Do you want to give it?
Because they usually have full valence electron rings, they aren't looking for more electrons and therefore are more stable and are less active.
Yeah, that's like the chemistry answer. What you guys can think about is like molecules have energy in a system. Like if you think about it, if you have a molecule that's bouncing around the room, it's not stable, right? It can give off energy. But if you have a molecule that's staying still, it has no energy to give off, right? So molecules that are also bigger in nature, an ATP versus an ADP. It has 3 phosphates, so it has a little bit more potential energy. And so breaking that phosphate bond can release energy. So that's what it means by more stable molecules have lower potential energies, if that makes sense. Carbs, fats, proteins all have high potential energies because you can break down the bonds between them to release energy. And once those bonds are broken and energy is released, those will be more stable molecules and at lower potential energies. Specifically, catabolic and anabolic are the words you should remember. Catabolic is oxidative and exergonic. Exergonic means what? Good. And yeah, catabolic, breaking down. Anabolic, building up. I think I mentioned that yesterday as well. So when ATP is used as energy, simple molecules with low potential energy can be used to make larger, more complex molecules. So this is the idea of coupling reactions. Does anybody know, does everybody understand why reactions are coupled?
Can you explain the difference between coupling and redox?
Redox reactions are coupled. That's true. Redox is like, you know Leo and GER? You know those acronyms? Oil rig also works?
No.
Okay. Leo, the way I remember it, Leo and GER are the two 3-letter acronyms. So loss of electrons is oxidation. LEO, loss electrons, oxidation. And then GER, gain electrons, is reduction.
Wait, why don't you just say they're the opposite?
Yeah, they're opposites of each other. But to remember the word reduction and oxidation, that's what you can do.
Like, redox sounds like reduce, but just think that biology's messed up, so it's the opposite. So reduce is actually you're gaining. So redox, you're gaining one, and then
redox is like reduction and oxidation combined. So
yeah.
All right, yeah. I'll get to that in a sec. Is everybody good for this slide? Very cool. So, once again, endergonic and exergonic reactions are relevant just because you can couple them. Coupling them means you can take an exergonic reaction that releases energy and use that energy to feed into an endergonic reaction that requires energy. So in cells, in anything, things that require energy, you don't just like get that energy like from random spots in the cell. It's like combining something that releases energy for something that needs energy. So breaking down ATP releases energy, and that can be used to build something up. And it could be whatever you want to build up. Building Legos, if you want. Does everybody get that kind of thing? Does everybody get why they're coupled? Cool. Can someone tell me which reaction would be spontaneous or favored? Yeah, why?
Because they produce products that have a lower energy.
Yep. And so would you say that the reactants are at a higher energy state or a lower energy state? The reactants, yeah, the higher. So the products are at a?
Lower.
Perfect. Perfect. Cool. Yeah.
These reactions have to happen like together, right? They can't one can't happen without the other.
Yeah, so technically the exergonic could happen without the other one, but the endergonic definitely requires the exergonic. Okay. Okay, so I'll quickly go over redox again. I don't have a slide on it, but I can kind of talk about it. Redox reactions are basically transferring electrons from one thing to another. So if you guys, we'll talk about NADH in a bit, but NADH goes through a lot of redox reactions with other things to basically transfer its electrons. That's how a lot of processes in biology works. You transfer electrons, you transfer energy. You can kind of think about it that way.
Are you losing an oxygen in redox reactions? No.
Not necessarily all the time.
But sometimes?
You could. But usually oxygen stays in the main molecule. You end up losing like a hydrogen usually.
Oh.
Yeah.
Ion, right?
Yeah. Cool. All right. So for metabolism, you need to know glycolysis. You don't need to know any of the intermediate names, enzymes, whatever you have it, but you should know what goes in and what comes out. Glycolysis, Laura split it up into 2 phases. You can think of it as 2, you can think of it as 1, however you like. Basically, 2 ATP is invested into the cycle. Invested meaning you have to get 2 ATP from the cell and put it into glycolysis. Even though glycolysis results in more ATP coming out. You have to first put a little bit of ATP in. So there is a negative feedback mechanism in this investment phase. When glucose is phosphorylated and one of the intermediates is formed, specifically fructose 6-phosphate. You don't need to know the name, but I'm just going to say it. When that is made, it's inhibited by ATP at high levels. Basically, this means if there's a lot of ATP around in the cell, the enzyme that makes that big name molecule is inhibited. Why would an enzyme that allows glycolysis to continue be inhibited if ATP is at high levels? Yeah.
If the metabolism is responsible for creating ATP and there's a lot of it in the cell, then it'll be a negative feedback loop because it'll inhibit the enzyme that furthers ATP production. So it's just the product regulating its pathway.
Yeah. The cell always wants to be as efficient as possible. So if there's already enough of the product in the cell, it doesn't need to make more, and so it'll inhibit it if there's high ATP levels. Another important part about the investment phase, phosphorylated glucose can't leave the cell. So this is kind of like the point of no return in the G1 checkpoint, but in glycolysis, once glucose is phosphorylated, it's committed to glycolysis and will be carried out fully. So that's another important point you should remember. But to talk about the payout stage, the investment stage splits the sugar, glucose, into 2 different molecules. Another phosphate's added to both of those molecules. This is not from ATP. ATP is then produced from a series of reactions. And then now, when ATP is synthesized from a phosphate group and ADP, what kind of phosphorylation do you call that? Yeah. What kind of phosphorylation do you call it when ATP is made from ADP and a free phosphate? You don't know? Does anybody want to guess?
Substrate phosphorylation.
There you go. Substrate level phosphorylation. And yeah, 4 ATP is produced at the end of the payout, which means how much net ATP is produced? 2. Correct. The net output is 2 ATP, 2 pyruvate, which is the 2 molecules that glucose splits into, and also 2 NADH. So you need to know all 3. Yeah. So before we get into the Krebs cycle, there's an intermediate step as those pyruvates are entering the mitochondrial matrix. 2 more NADHs are made as those pyruvates become acetyl-CoA. And acetyl-CoA is the main molecule in the Krebs cycle. That's all you need to know about acetyl-CoA. Cool.
So for pyruvate oxidation, it's 2 pyruvate is the input and the output would be 2 or 4 NAD?
2 NADH and 2 acetyl-CoA. So 1 for each.
Okay.
Okay, Krebs cycle. You don't need to know any of the intermediates. That's beyond the scope of this course. But you should memorize the fact that there's 3 NADH molecules, 1 FADH2. It's just a different name, but it pretty much does the same thing as NADH. 1 ATP, again, substrate level phosphorylation. And 2 carbon dioxides are made in 1 turn of the Krebs cycle. And because you have 2 acetyl-CoAs, Krebs happens twice. So basically, those products are now doubled, which you have to remember. So in total, 6 NADH, 2 FADH2, 2 ATP, and 4 CO2. That comes out of the Krebs cycle for one glucose molecule. Yeah.
On the exam, do you think she's gonna refer to it as the citric acid, or we can just remember that it's only Krebs?
It's both are the same thing. You can say either.
What if we forget? Do you think she's going to use citric acid, or it's just gonna be Krebs?
She could honestly interchange it. Yeah. So yeah, again, it's also called citric acid cycle. So make sure you can remember that both of those terms are interchangeable. Okay. Lastly, electron transport chain. If it's not substrate level phosphorylation, it's... What's the other type?
Oxidative phosphorylation?
Yep, that's correct. Lot of information here, but it's all important. So make sure, I'll go slow, but make sure you have it down. This happens in the inner membrane of the mitochondria. NADH and FADH2 have electrons that they dump into that chain. So again, electrons, you can think of them as carrying energy. And NADH and FADH2 are emptying their buckets of energy into that chain. Again, redox reactions happen here. NADH and FADH2 are both oxidized to NAD and FAD. And in turn, if something's oxidized, something else has to be reduced. So in this case, complex 1 is reduced because it gains the electrons from FADH2 and NADH. So the chain reactions between all the complexes send all the electrons through to the end. What's the final acceptor of all the electrons? Oxygen. The potential energy of the electrons drops at each step. Do you know why? Or how, I guess? You can take a guess.
I was gonna say like the electrons are being like given out of the molecule, so the potential energy of the molecule would grow.
Yeah, that's very close. Yeah, I think I would accept that as an answer. All I would say is that it starts at the beginning of the chain when each of the complexes is wanting those electrons, and as you go, there's one less complex, and yeah, there's one less complex each time that's kind of pulling for those electrons until you get to the end. But the energy in that system is used to do the work being pushing the hydrogen ions into the intermembrane space. Can someone tell me why there's a gradient? Or what purpose does the gradient serve? Yep. Yep, that's correct. So there exists a gradient because once you have a large pool of hydrogen ions, those can then flow down the gradient. You don't need energy to do that. It'll flow by itself, cause ATP synthase to spin and make ATP. That's the extent of what you need to know about that. And finally, at the end of the chain, where do the electrons go? As you guys said before, it's oxygen, which then combines with the protons to make water. Is that clear to everybody? Can I get thumbs up from everybody? Sweet.
How many ATP are produced in UTC?
It's like 32
32?
total in all of, yeah. Let me get back to you on that. Yeah, I forgot the number off the top of my head.
How much ATP is produced?
In the electron transport chain, yeah. Okay, so I did not cover fermentation on purpose because I want one of you guys to talk about it. What is the point of fermentation? Sorry. One more time.
So energy can be produced like in an anaerobic system?
Yep. Perfect. And why does it matter that it's anaerobic?
Because anaerobic means that there's no oxygen present. So if we didn't have oxygen, that would be the only energy source.
Mm-hmm. And then the final question: what is the purpose of oxygen in the whole system?
It's the final electron acceptor.
Yep. Perfect. So oxygen is the final electron acceptor. If you don't have oxygen, it's kind of like there's no point in the Krebs cycle or the electron transport chain from happening. It's like the main driver of those two processes in the end. So without oxygen, those two can't happen, just glycolysis. But you need to replenish the NAD in glycolysis because glycolysis makes a lot of NADH. So fermentation regenerates NAD+, and like you said, energy production in anaerobic conditions is why. Can somebody tell me what the two types of fermentation are?
Lactic acid and alcohol fermentation.
And which happens in which? Or,
Doesn't
yeah.
alcohol happen in lactic acid? Or is it the other way?
Or sorry, which? All right, I guess the question is, which happens in plants and which happens in humans?
Lactic acid's in humans, not because we make it when we exercise.
Yep. And then alcohol is in plants.
It can happen in humans too, autourinary syndrome.
That's true, but it usually doesn't happen.
Always drunk.
All right, that's fair. You want another piece of candy for that?
No.
Okay. That was good though. That was good. All right. Any questions? Topic 9. It's topic 9, right? Yeah.
Can you tell, like, steps of lactic
Gotcha.
acid and alcohol?
What's that? What's that?
Do we need to know the steps of lactic acid and alcohol?
No, you don't. You just need to know that it regenerates NAD+.
Oh, 30, 32 ATPs produced.
Oh yeah, 30 to 32 ATPs produced in the electron transport chain.
Is the reason why we die so fast without oxygen is because the lactic acid builds up so fast?
No, glycolysis can't. I mean, what's the exact reason? You need oxygen to go to your tissues, so I think that's faster. Without oxygen in your tissues, actually it is pretty much this. So without oxygen, it can't make enough energy, and so if glycolysis were to just keep running, it's not enough. I think that's probably a better answer. Yeah.
Doesn't the buildup happen when you die though, and that's why you like kind of shake?
Yeah.
Yeah.
Well, you shit yourself because that's what happens when your body loses control of it.
Oh yeah.
Yeah.
How about you inflate and then?
Maybe. I don't know. I don't really know. All right. Any other questions on topic 9? Cool. All right. Topic 10. This is the meiosis, non-disjunction, epigenetics topic. Who wants me to run through meiosis in like a decent amount of depth? Raise your hand. All right, cool. I will. So, I took this from my topic 10 review session, but meiosis is essentially going from 2n, one cell with 2n, to 4 cells with n. I have some old drawings here, but yeah, just wondering if I should. Okay, no, we'll keep it. Okay, so quickly to go through all the parts of meiosis, you have prophase, metaphase, anaphase, telophase, and then repeats again for meiosis 2. Slight difference in meiosis 1 and meiosis 2. You start with prophase 1, which condenses the chromosomes. The homologous chromosomes pair up. So homologous chromosomes, if you can see this, is when you have both of them. Basically, you have a set. Crossing over occurs. Does everybody understand crossing over? Does everybody understand why crossing over happens? No? Okay, crossing over happens. It's just a process that happens in cells. You don't need to know how, but essentially, little bits of each chromosome flip-flop from one to the other. This is how genetic diversity is increased, because you get one from your mom, one from your dad, and each allele for each gene could be slightly different. Before I continue, does everybody understand the difference between alleles and genes? Thumbs up? No, okay. Alleles, the difference between alleles and genes are everybody has the same set of genes, but each gene has a different counterpart, which is called an allele. So if you guys talked about like big A, little a, those are two different alleles. Alleles is just like a slight mutation, not a mutation, a slight difference in gene in one transcript versus the next. And you get one allele from your mom and one allele from your dad. Yeah.
When you say big H and little h, are you talking about like the crossing over, like the homozygous and heterozygous?
You think of Punnett squares? Like the
Yeah.
box? And, yeah.
Is that okay?
Yeah. Does that make more sense?
Yeah.
Okay, cool. So crossing over occurs, and then metaphase, all you know is that they line up in the middle of the cell, and anaphase, they split apart. Yeah, so the homologous chromosomes split apart in meiosis, in anaphase 1, which causes the chromosome number to go down by half, and what you're left with is a set of sister chromatids. So does everybody get the difference between homologous chromosomes and sister chromatids? Yeah? Cool? All right. Important thing to remember, after meiosis 1, it is haploid. They're no longer diploid. So when you start meiosis 2, the chromosomes will once again condense. They'll line up at the center of the cell, and then split again, but this time the sister chromatids themselves will split. And then this will result in 4 different cells that are all unique, like completely unique. And these are the haploid gametes. Everybody cool on that? Yeah.
So in meiosis 1, would they all be diploid?
Before meiosis 1, yeah.
Okay, so meiosis 2, they're all haploid cells?
Wait, sorry, one more time?
So in meiosis 2, they're all haploid
Yep.
cells? Okay, thank you.
Yep.
What's the product of meiosis 2?
4 haploid gametes from one cell at the beginning.
But they're still considered haploid in the beginning?
So the very beginning, before meiosis has started, they're diploid, 2N.
Yeah.
After meiosis 1, even though the cells look like this, these are haploid.
Okay.
Because their other set is in the other cell, basically. Does that make sense to people?
So it goes from 2N to what?
Yes.
To what?
Oh, 2N to just regular N, just N.
Oh, okay.
Yeah.
Or 1N.
Or 1N, yeah. There you go. You want candy? Yeah. All right. Everybody good on meiosis? Cool. Yeah, main things to remember. Crossing over, haploid, and sister chromatids all after meiosis 1. Okay, non-disjunction. You guys have all seen this chart before. Non-disjunction can happen in both meiosis 1 or 2. Which one is more serious, do you think?
Meiosis 1.
Yeah, why?
Because it's just earlier in the stages, so its effect is going to be much bigger on the end of the product.
Yep, it's going to have an amplified effect later on. Yeah, so meiosis 1 is when the homologous chromosomes don't separate. And then meiosis 2 non-disjunction is when the sister chromatids don't separate. Here are a couple of the examples of what happens when non-disjunction happens. You don't necessarily need to memorize every single one. You can just remember trisomy is an extra chromosome. Monosomy is one less, or just one chromosome copy. And polyploidy is more severe, more complex, just a whole bunch of crazy stuff. All right. Any questions on non-disjunction? Yeah.
So the product of non-disjunction in meiosis 1 is 2 diploid and 2 non-viable, and the product of non-disjunction in meiosis 2 is 2 haploid and 2 non-viable.
Yes. That's correct. Okay. Another big part about topic 10 is chromatin. What are we doing on time? Still looking. Chill. Okay,
Yeah, we're
chill.
good.
We're chilling.
Wait. Yeah.
Okay, so chromatin is your whole DNA, histones, other proteins. It forms nucleosomes. Like chromatin is like kind of everything. You think about it as like everything. And the big thing that you should remember is that how it's bound affects transcription. So whether it's more tightly bound or more loosely bound, that affects transcription. Open chromatin is loosely bound. It's called euchromatin. And it makes sense because if chromatin's less packed together, transcription factors can access it. And so RNA polymerase can access it too, transcription can occur. But if chromatin's closed, or it's called heterochromatin, it's tightly packed. And if it's tightly packed, transcription factors can't access those sites. So gene expression there is silenced because transcription can't happen. Does that all make sense? Cool. Okay, so also important are histones and how they're modified. You can have histone acetylation, which generally causes open chromatin. So you add an acetyl group to histone by that enzyme, histone acetyltransferase. It'll add the acetyl group and promote open chromatin. The opposite function of acetyltransferases are histone deacetylases, and it basically reverses what the other enzyme does. It removes the acetyl groups, which promotes closed chromatin. And closed chromatin means transcription can't happen. The other type is histone methylation. It doesn't change the charge the way acetylation does, but it can have different types of effects. You don't really need to know like how exactly, just know that methylation can cause open or closed chromatin. And then a couple extra facts just for your own knowledge. Lysines can be mono, di, trimethylated, and depending on how it is, it can be either open or closed, but they all have varied effects on gene expression. So if Laura basically asks you a question on methylation, you can say, depending on how it's methylated, if it's multiple methylated, it'll either be open or closed. That's kind of all you need to know for that. And then lastly, epigenetics. It's a way to describe how gene expression can be different by something other than the genes themselves. So you guys know how mutations and transcription factors can affect expression, but epigenetics represents how open and closed chromatin can also affect expression. So the fact that that chromatin can be open or closed, that state can be passed down through generations. So if you have the trait for eye color, and it happens to be more closely packed in your parents, that can be heritable. And you can inherit that closely packedness around the gene of eye color, if that makes sense. Cool? Cool. Yeah, epigenetics means above genetics. That's kind of like the definition, or like the breakdown of the word. Yeah.
So is histone modification post-translational?
Yes. Because you're adding something to the already formed histone. Cool. That's kind of all we had for slides. Any questions for topic 10 or anything?
Well, we need to know about histone phosphorylation.
It's another type of modification. You won't. Let me get back to you.
Okay.
Yeah. I don't think you'll need to know specifically what the effect is. You can just know. You just need to know that it happens.
Okay.
My guess is yeah.